90 degrees C for 2 hours, and replicate 3. Okay, and then the second one, after I've done with this one, you need to do 80 degrees C 1 hour for rep 1, and so on and so forth. Okay, so the way that you can list it, okay, is by using this table. Okay, there's no standard for doing this, but this is just a suggestion. Okay, but you are free to do how you deem fit. Okay, as long as you know you have your run order, and then you follow your run order. Okay, so for example, run order 1. Okay, factor 1, for example, if you were to take factor 1 as the temperature, so this is the temperature and this is the time. So factor 1 temperature will be 90 times 2 hours, replication 3. And then for the second run order, okay, you will have 80 degrees C 1 hour, replication 1. And then for the third one, okay, you will have 80, okay, and 1 hour, replication 3, and so on. Okay, so now you have your run order, okay, established. Then when you go into the lab, then you need to follow this run order. So now you have your replication and you have your randomization. Okay, okay. Okay, so another type of factorial design is what we call as factorial design with blocks. Okay, so as we have discussed previously, then we do blocking because we are concerned that there may be confounding factors, or we really know that there are confounding factors that may create bias, okay, onto our experimental samples. Okay. Okay, so what we want to do is to create blocks so that within each block, okay, everything is uniform except, everything is constant except the factors. Okay, so between the blocks there may be differences, but within the blocks, okay, everything is the same except the factors. Okay, so what we need to do is, okay, so usually, okay, if it is if it has certain limitations, for example, like time, sample availability, etc., so that you are concerned that it may affect, okay, may confound, okay, the results of your experiment, then you it would be advisable for you to create blocks, okay, for your experiments. Okay, so when we create blocks, so basically when we randomize, we did not randomize all the treatments and the replications. Okay, so there will be randomization, but there will be limited randomization where the randomization will only occur within each block. Okay, it doesn't occur among all the samples. Okay. Okay, so it is similar to CRBD, completely randomized block design. Okay, so there is randomization, but only within the blocks. Okay, and then usually for food science experiments, okay, it is convenient for us to take the replications as the blocks. Okay, because first of all, then we will only have to deal with a fraction of the number of samples. Okay, and then secondly is that we will be able to capture the heterogeneity of the samples, because we are not taking all the samples at one time, then we are taking it from different batches or at different point of time. Okay, so then we will be able to incorporate, okay, the heterogeneity, the differences of the samples, okay, into our results. Okay, okay, so because so in food science, okay, most of the time you are dealing with situations in the lab. Okay, so it is more convenient, okay, for us to be able to do our experiments so that we can finish it, okay, within a certain amount of time that we are convenient with, and also it will not affect the samples. Okay, for example, we cannot finish it today, then we have to store it, and we are not sure that whether the storage will have an effect, okay, on the properties of the samples or not, that may in turn affect the results of the experiments. Okay. Okay, so if you were to take the replication as our block, okay, then what we need to do, okay, is to do the first rep first. Okay, so we will block the replication, so we will only do the first rep, okay, and then after that the second rep second, and the third rep third. Okay, and there are no randomization between between the blocks. Okay, there are no randomization. Okay, so if you were to look at this example over here, okay, so if we are going to do this the same experiment, but now because we feel that to manage 27 samples at once, that it is going to be very tedious. Okay, and you are concerned that maybe it will affect the outcome of our experiment. So what we can do is to create blocks. Okay. Okay. So let's take, for example, we want to take the replication as the block. So meaning that we will have 3 replications, meaning that we have 3 blocks. Okay. So when we do our experiment, we will do the first rep first. So that is the first block. Okay. So meaning, if as opposed to having to deal with 27 samples, so now we are only dealing with 9 samples, because these are in one block. That is the first replication. Okay, so that will cut down the number of samples that we have to deal with dramatically. Okay, from 27 to 9 samples, which is more manageable. Okay, it is more manageable. Okay, so how do we do this? Okay, so for example, we have the same experiment. We have 2 factors, that is the temperature and the time, and we have the levels. For temperature we have 3 levels, for time we have also 3 levels. Okay, so for treatment, okay, now we have, it is combination of the levels. We have 3 times 3 equals to 9 treatments. Okay, so these are our treatments: 71, 72, 73, 18, and also 90. Okay, so we have 9 samples. Okay, so now we are going to do the first rep. Okay, so we have we go into the lab. Okay, so meaning that we only have to prepare 9 samples only. Okay, so before we go into the lab, we need to establish our run order. Okay, so how do we establish our run order? So for example, if we are going to use the full straw method. Okay, so now what we need to do is to prepare 9 pieces of paper. Okay, on each piece of paper, so we need to write 71, and then second one 72, 73, and so on and so forth, until we have 9 pieces of paper. Then we roll them up, okay, and then mix them, and then pick randomly. Okay, so if you were to do that, okay, for example, when we pick randomly, then we get 80 degrees C for 2 hours, and then second one 90 for 3 hours, and then 70 for 1 hour, and then we got 70 for 3 hours, for example. Okay, and so on until we finish we have our 9 treatments. Okay, so this will be our run order. So meaning that when we go into the lab, okay, we need to prepare 9 samples, and then for the first sample we will subject it to 80 degrees C of heating for 2 hours. Okay, and then for the second sample we will subject it to 90 degrees C for 3 hours. Okay, so how do we list it? Okay, so this is just a suggestion. This is one way of doing it. Okay, so we have, this is the run order, and then this is the replication, 1, 1, 1, 1. Okay, and then 2, 2, 2, 2, and then 3, 3, 3. Okay, and then we have the first factor, and then the second factor. Okay, so for example, if this is our run order, so the first factor, this is the first replication. Okay, for the first factor, we have 80 degrees C, and the second factor, it is 2 hours. 80 to over here, The second run order, it is 90 degrees C and 3 hours. Okay, and the third one is 70 degrees C, okay, and then 1 hour. Okay, for the fourth one is 70 degrees C and then 3 hours, okay, and so on and so forth. Meaning that if you have 9 samples, then there will replication 1 will have 1 until 9 samples. Okay, after that, we prepare for the run order for the second replication. Okay, so for the second replication, we should not use the same sequence. Okay, do not use the same sequence because you have to randomize it all over again. Okay, so for the second treatment, okay, you still have to prepare. You can use the 9 pieces of paper, okay, and then you mix them up again, okay, and then you just pull it. Okay, so for example, if this is for the second rep, R2, this is for the R1, okay, and then for example for R2, okay, for 1 you pull 1 piece of paper, you will get 70, okay, and then 3 hours, and then you get 90, 2 hours, and then you get 90, 1 hour, and then you get 80, 1 hour, for example, okay, and so on. So this will be the run order for the first rep, this will be the run order for the second rep, and similarly you have to do it again for you to get the run order for the third rep. Okay, so when you go into the lab, actually the first time you only do for the first time you only do for the first replication. So you follow this run order and then finish everything. Okay, so maybe you want to analyze it first, still it is okay, or you want to store it first, that is also okay, as long as you are confident and you have proof that when you store in that storage condition, okay, it will not affect the properties that you are going to measure. Okay, for example, it will not affect the DPPH value. Okay, and then after a week, when you have finished everything, you want to do the second replication, then you will follow this run order. Okay, and similarly for the third replication. Okay, so for both design of experiments, okay, remember the objective for us is to determine the effect of the factors. We want to determine the temperature, and also we want to determine the time. Okay, so for both design of experiment, okay, we in order for us to analyze it, determine the effect of the factors, then we need to do ANOVA, analysis of variance. Okay, if it is a if it is a one-factor experiment, then definitely we need to do a one-way ANOVA. However, if it is a more than one factor experiment, okay, then we have the option of using a two-way ANOVA. Okay, if you were to use a one-way ANOVA, then that ANOVA will tell us that whether there are significant differences or no significant differences between the means. But ANOVA will not tell us which mean is significantly different, okay, from which mean. Okay, so in order for us to determine which means are different, then we need to do a multiple means comparison test or a post hoc test. Okay, so if it is only 2 levels, then we can use a t-test, but if it is more than 2 levels, then we can use Duncan, Fisher, or Tucci's. Okay, so if another option is for us to use a two-way ANOVA. Okay, for us to use a two-way ANOVA. Two-way ANOVA, okay, will tell us statistically and objectively whether the whether the factors have a significant effect or not. Okay, so for example, in this example, the two-way ANOVA will tell us objectively, okay, whether temperature has a significant effect on DPPH or not, and time has a significant effect on DPPH or not. Okay, And also the advantage of using a two-way ANOVA is that it can tell us, okay, whether there are significant interaction between temperature and time or not. Okay, so it is more advisable, okay, to do a two-way ANOVA as opposed to a two-time one-way ANOVA. Okay, so what do I mean by a two-time one-way ANOVA? Okay, sometimes when we read papers, we can see that there are a table of data, and the way that they write the data is like, for example, this is time, okay, 1, 2, 3 hours, for example, and this is the temperature, okay, degrees C, 60, 70, 80, for example, and these are all your data. And then you can see that there are two types of notation over here. One is capital, one is small letter. Okay, meaning that, and then